with this, Karim, the stage is yours, and thank you once more. Afterwards, please, we have a question and answer round. Post your questions in the chat. I will then accordingly read them. And yeah, that's it. Thank you very much.
Yeah, so thanks a lot for your kindness to Felix and Daniela for allowing me to talk to all the students, explaining them what I do at my job every day, and also what our company does. So, little bit about. Can you please confirm, Felix, that you see my screen?
Everything is perfect. Go on.
Okay, super. Thanks a lot, Felix. So yes, I have finished. My name is Karim Vadiwala, and I finished my PhD in molecular biology at Max Perutz Laboratories about 2 years ago. And since then, I've been working as business development and sales manager at EUCODIS Bioscience, which is a biotech company based on campus Vienna Biocenter in Vienna in the 3rd district. And recently, we've been acquired by a Swiss company called Bioscience. That's why you see in our new logo, EUCODIS, which is part of now Swiss Life Science Bioscience group. And again, thanks a lot, Felix and Daniela, to give me an opportunity to present what my job role involves at EUCODIS Bioscience. I would first like to explain what we do at EUCODIS Bioscience. So a little bit about our company. We are enzyme engineers, and we have developed proprietary toolbox for enzyme optimization that includes in vivo recombination, gene shuffling, and other mutagenesis approaches. And since past 15 years, we have used this proprietary toolbox for enzyme manufacturing and used these tools to manufacture about or more than 50 well-characterized enzymes that we routinely supply to pharmaceutical companies, cosmetic companies, diagnostic companies, fine chemical companies globally. We are a very global company. We have customers globally, including parts of Asia that includes Japan, China, India, Pakistan, Bangladesh, Middle East like Dubai, Gulf, Egypt. And then we also ship our enzymes to America and different countries in Europe. So we are a very global company. And in addition to our commercially available off-the-shelf enzymes, we also have customized protein manufacturing services. So we do microbial fermentation and to produce enzymes, and we do every step of the value chain. That includes process development, scale up, and other parts of routine protein manufacturing. We were founded in 2007, so we are about 15 years old, and we are headquartered in Vienna, Austria. And we are ISO 9001 certified company. That means all the enzymes we manufacture, they are produced and manufactured under ISO 9001 grade. Now, moving forward, so besides our enzymes and customized protein manufacturing services, we also have some technology. So I will just shortly explain one of the key interesting technology that might be interesting for some of you. So this is a technology that we have developed and patented for tagging different kind of proteins and antibodies with drug molecules or fluorescent molecules. So what you see here is a cartoon of recombinant antibody in the blue color that has a short linker peptide on the left side of the screen. And then we have generated an engineered protease and enzyme that comes and recognizes this linker peptide and helps to attach or conjugate this recombinant antibody with a drug molecule, generating an antibody drug conjugate. So just to explain you briefly, such antibody drug conjugates are used by pharmaceutical companies for clinical trials to inject in human patients for delivering the drug at the site of your interest, at the tumor of your interest in the patients. Therefore, this technology can be used to cure cancer. And in addition, it can be also used to detect different kind of markers in the diagnostics or in also in imaging field. So you can also use it for detecting different kinds of molecules of your interest for antibody labeling, for example. And we are looking forward to out-license this technology to different pharmaceutical and diagnostic companies. Now, I would like to briefly mention also about Bioscience. So this is the Swiss life science company that has bought our company in Vienna, EUCODIS Bioscience, and a little bit about Bioscience. So Bioscience is a very global life science provider and a chemical manufacturing company. We have a peptide and peptide manufacturing units in America and Netherlands, and then we have chemical manufacturing plants or units in Slovakia, Switzerland, England, and China. And then we have an antibody manufacturing facility in Dublin, Ireland, and we are the enzyme manufacturing site of Biosynth in Vienna, Austria. Yes, so we are very global life science provider and manufacturing company for different types of chemicals. And now I would like to briefly mention about myself. So about my education and my experience. So I have studied my bachelor's in biotechnology at University of Pune in India. After which I've received a Swiss scholarship from a foundation in Geneva to study my master's at University College London, and I finished my master's in prenatal genetics and fetal medicine at UCL London. And as part of my master's, I also went to Cambridge at Bebraham Institute, Cambridge, to finish my thesis research project on placental stem cells and epigenetics. And after this master's, I worked as a research assistant or a technician at a neuroscience lab in UCL London. In London. And following this work and a little bit of research experience, I came to Vienna to pursue my PhD at Max Perutz laboratories in molecular biology and also neuroscience at University of Vienna in Vienna. And since about past 2 years after finishing my PhD, I have joined EUCODIS Bioscience, and I've been working as a business development and sales manager. And before I actually go into business development and sales role to explain what exactly I do on day-to-day basis, briefly I would like to explain you all what I did during my PhD at Max Perutz Laboratories. So at Max Perutz Laboratories, I have published, along with other co-authors, I have published a paper in a neuroscience field in Science Advances. So this is a technology in the field of tissue clearing, labeling, and imaging. We have developed a method called Deep Clear that enables to make different kinds of organisms completely transparent like a glass. Then you can image them after labeling them with different kinds of antibodies and RNA detection methods. So what you see on the screen is a Z-stack recording of a zebrafish that was first pigmented with black melanin pigment in the eyes. And with our technology Deep Clear that you can study and refer to in the article in the Science Advances in 2020, what you can see that we have made the fish completely transparent. We have made it pigmentless using our technology Deep Clear. Then we have labeled them with an anti-acylated tubulin, labeling the entire brain nerves and eye nerves. And you can thoroughly image it with a light sheet microscope. And without the need of sectioning the tissue, you can study the entire fish labeled with this neuronal marker in about weeks' time or 2 weeks' time, rather than taking years of time. Yeah, because you don't need to do any tissue sectioning now. Now, coming back to my role, what I do at EUCODIS Bioscience. So I am a business development and sales manager at EUCODIS, as I mentioned in the first part of my talk. So one of my key responsibilities is to do business development at international trade fairs. So what I do is, for example, just to give you a glimpse of my year that I spent in 2022, this year at EUCODIS, I have visited the following fairs internationally this year so far, and I will be visiting some more. So yeah, one of my key duties or responsibilities is to visit such trade fairs internationally and talk to our customers to understand what kind of enzymes they need from us, and we could offer such products to these customers. And in case if some of our enzymes that we offer them does not fit into their portfolio or projects, then which kind of enzymes we could develop for them in a customized fashion, for example, as a R&D project. So these companies, pharmaceutical, cosmetics, diagnostics, or fine chemical companies would pay us to develop such customized enzymes, and we would develop for them in a customized project. That is what I do at these trade fairs internationally. So now I will walk you through briefly which fairs I have been recently in this year, so that you get a general idea that, yeah, what my role involves. For example, in the beginning of the year in April, I was at this fair called in Cosmetics Paris. And as you know, France is very famous for its cosmetic companies. And in addition to the famous French cosmetic companies, this fair attracts globally relevant and very reputed cosmetic companies from countries including Japan, South Korea, India, China, and also different countries in Europe, including Switzerland, England, and parts of America. So I have spoken to many clients and customers who are interested in using our enzymes for producing cosmetic ingredients that can be used in cosmetic lotions and novel cosmetic products. Now, going forward, next I was at an international fair called Bio in San Diego, California in June. And this Bio fair is known to attract international pharmaceutical and biotech companies from entire globe. So around 15,000 people came from entire planet to attend this fair for 3 to 4 days. And I have spoken to especially companies from Japan, South Korea, and different countries in Europe who have shown interest in our bioconjugation technology that can be used to tag different kind of antibodies to generate antibody drug conjugates that can be used to cure cancer. So these companies from South Korea, Japan, and from different European countries have shown interest to buy our technology, or to say to in-license our technology, yeah, which can be used to cure cancer. Going forward, in September, I was at a trade fair called MedFit, and it's a very reputed trade show in the French biotech cluster. And the main focus of this event is diagnostics. So they attract many diagnostic companies who aim to develop diagnostic tests for human diagnostics for detecting different kinds of human diseases. And I spoke to a lot of clients at this MedFit event who are interested to use our enzymes in the field of human diagnostics to detect different kinds of genetic diseases. And lastly, I would like to explain you, I was at this fair recently called Nordic Life Science Days in Malmo in Sweden. And at the main focus of this event is mainly Nordic bioscience and biotech companies that come from the Nordic region, that includes Sweden, Denmark, Norway, Iceland, and Finland. And I have spoken mainly to companies that are interested in developing single cell technologies, and different types of diagnostic companies that are interested in using our enzymes for single cell technology preparation and also for diagnostics. Yes. So I'm in touch with such companies that I spoke to at Nordic Life Science Days. And now I would like to explain you that what is my key role in EUCODIS Bioscience. What do I do when I work at EUCODIS? So for example, as I told you, one of my key role is to do business development, to talk to customers and to gain new customers by explaining the applications of our enzymes and which kinds of enzymes we can manufacture for them on a customized basis. So we can manufacture up to kilograms of enzymes for different kinds of customers, and that is what I do in business development. And in addition to that, I also do marketing. So I do marketing on different social media platforms like LinkedIn, that you are all familiar with. And then I also do marketing on different kinds of pharmaceutical marketing platforms like there's a British platform called Pharma Industry Review. I generate some kind of content for my company on this Pharma Industry Review platform. And in addition, I also generate marketing flyer for my company, enlisting our products and services and technologies. So recently I have finished with a Slovakian company, marketing company. I have generated a content or a marketing flyer enlisting our different kinds of products that are used in pharmaceuticals, in diagnostics, in vaccine manufacturing, and also enlisting our services and this conjugation technology that I explained you. Then I also do price negotiation with customers. So we send customers our offers for products and services. And especially this price negotiation is important in markets, price sensitive markets like India, Pakistan, Bangladesh, where customers really negotiate even on cents to save their cents and euros. So I negotiate with such customers, trying to offer the best offer for us to create win-win situation for the customer as well as for our company. And then I also do cross-team collaboration with the scientists. Whenever we have new projects for different kinds of customers, I interact on day-to-day basis with the scientists and technicians in my company. And what does my daily routine or day look like? For example, if a customer sends us a request for different enzyme, and if we have this enzyme, then the normal procedure is that I will send them the quotation or offer explaining how much this enzyme costs. But it also happens usually on day-to-day basis that certain enzymes that customers need, we don't have such enzymes. So in those cases, I need to interact with my scientists and ask them if we can produce this enzyme in what time frame, what would be the cost, and how we could deliver such enzymes to customers. And then I also interact with our finance team and ask them exactly, what price we can offer such offers to our customers. And then before shipping the product to the customer, our quality control and quality assurance team at EUCODIS Bioscience checks and confirms if the product meets the criteria that we should be fulfilling the criteria or the norm that we are used to as a company, as a ISO 9001 grade company. And in addition to my daily routine that I explained you, we also have consultants that help us to grow our business in different international markets. For example, Mr. Blanc, he helps us to gain new customers in the French cosmetic markets because he's French and he speaks French and he has a very good know-how and knowledge about the cosmetics industry. Then we have Mr. Michael Hansel in Austria, and he has a very good know-how amongst clients in Austria and Europe and also China because he's an international dealer also in China. So he helps us to grow our business in Austria, Europe, and China. And then we have in the DACH region, Mr. Wolfgang Haufe, and he helps us to grow our business in DACH region in Germany, in Austria, and also parts of Switzerland. So I also interact on weekly or monthly basis with our consultants. And in addition, we also have distributors in international markets that include South Korea, China, India, Pakistan, Japan, Egypt in the Middle East, and America. And I interact and try to understand how we can expand using these distributors in these international markets. So who are distributors? Distributors are companies who first buy our enzymes and then resell our enzymes to their customers. So it helps to multiply our business because these distributors have existing customers. They first buy from us and sell our enzymes to their customers. And now I would like to explain you or some suggestions which are the key skills required for doing this job. So in my opinion, one is one needs to be very good in the communication. And so you need to first understand what the customer exactly needs, what are the customer requirements, what are the challenges that customers face in biotech, academics, in pharmaceutical companies and diagnostic companies and fine chemical companies and in cosmetic companies, and how we can assist them to develop these products and then deliver them on time. So communication is very important with the customers as well as within the company. And then I would say empathetic listening. You need to really listen what the customers need, as I told you. And then, as I told you, also in price sensitive markets like Asia, one needs to be really good in negotiation with the customers on prices. And then, as I told you, you need to do cross team collaboration with the finance team, with the research and development scientists, with the quality control and quality assurance team, and also with the customers. And I wrote one point: patience and perseverance. Why is this important? Because sometimes when you have a new customer, sometimes to close a sales deal, it can take a week, like few days, which is a best case scenario. But the worst case scenario is that sometimes it can take even up to a year. So you need to be really patient to deal with the customers. Now, going forward, some tips for being successful at this job. Yeah, I would say one of the key skills, people before joining such a job try to do MBA, which I don't say that it's a bad degree to have. I think it's good to have a business knowledge. But one of the very key important skill that after doing this job for 2 years, which I would like to recommend people who are interested in pursuing such a role in biotech, pharmaceutical, diagnostic, or other types of job in such a business development role or sales job. One very important skill to learn or have is to learn and understand different kinds of culture and people. For example, just interactions with people. So for what helped me, which I would like to friendly explain, I attended every week during my PhD. During while doing my PhD, there is a beer hour at Vienna Biocenter, which people might find funny, but I got to interact with international culture, international people from across the globe. I met people from Japan, China, India, America, Canada, Egypt, Turkey. And this gave me a good feeling how people interact with each other, what people want. Yes, so that you can develop trust with people. And people can also trust in whatever you say, the genuine connection between people. And then comes to business courses. As I told you, an MBA is useful, but it's not a must. So if you do some business courses, and if you have some next point, some business related internships or work experience in biotech, pharmaceutical companies, it can be easier to get such a job in different companies in the job market, in the industry. And then one key important aspect is to serve the customer and always stay customer focused. Yes, whatever customers need, try to have genuine interaction with customers regarding the delivery, whether you can develop such a product for them or not. And always over deliver, promise less, but over deliver to your customer, and never miss the deadlines, or try to at least not miss the deadline. And just some recent changes that we've had at EUCODIS Bioscience. As I told you, we have been recently bought by a very giant Swiss life science company that has also division in small molecules. They sell small molecules and they can also manufacture small molecules. They can manufacture peptides, different kinds of monoclonal, polyclonal antibodies, and we are the enzyme unit for this company. And recently, we have been encouraged also to interact with the sales units or business units of small molecule division, peptide division, antibody division, and such cross interaction or interdivisional interactions between different divisions of Biosyn. What I have observed is that this has promoted our sales. This has got us new customers. So it's a very good strategy that we are following currently. And just to mention briefly that some of you might be looking for scientist jobs. So our company has R&D positions open for different scientist roles as well as technician roles. People who have PhDs or masters in molecular biology and who are interested to join our company are very welcome to contact us on my following email. And also, any one of you who is interested to pursue such a role in business development or sales can contact me directly or contact Daniela and Felix, and I would be very happy to help them, to talk to them, to have a coffee and have a chat with them. Thanks a lot, Felix and Daniela, again, to give me a chance to talk to students on this platform.
Karim, thank you. It was very nice, very informative. I got some questions for you. Maybe you can tell us about your way to this position, and we had a chat before the session started, and also the participation of the GmbH regarding the CV checkup. I think this would be nice for our younger people here.
Yeah, so what I did, Felix, while I was finishing my PhD, what I realized that I had two passion. One was to do drug discovery, which I believe our next speaker, Dora, will kindly update the students. And the second passion was to do business. And the thing is, I was very confused. So what I thought, let's do some sort of internship and let's attend some business courses. So COVID came at that time, and for me, it was one of the fortunate times, because what happened is that during this time I started to write my thesis, my PhD thesis, and one of the business person or business friends from Boehringer Ingelheim very kindly introduced me to one business course that was offered by Boehringer Ingelheim totally for free. Such a business course usually takes about you must pay about 3 to 4,000 euro to get trained for business. But I think because COVID hit, they offered such a course totally for free to students and people who are interested to learn. So I joined such a course, and I did some 1 or 2 short internships at one biotech company in Austria and in America. And I really liked business. And then I asked myself a question. I was doing research in my laboratory anyway during PhD, and I published 3 papers during my PhD. And I asked myself a question: given a possibility, would I choose a path in business or R&D? And for me, it was very clear after attending such business courses and doing such internships that I would really like to talk to customers and not do R&D lab work. That's why I decided to then apply at EUCODIS Bioscience, and I had a very rigorous interview rounds, 2-3 rounds, and after clearing the 2 rounds, they gave me a case study to solve on a product that EUCODIS has, how to launch this product in the Asian market. So then I had to take 3 days to solve this puzzle, and they liked all my answers and they gave me the job. So that's how I made it to this job.
Okay. You answered a lot of questions already, also regarding the soft skills. And maybe one question remains. You said something about social media. What's the role of social media? How important is it? And what's required? Also, which skills are required in your job, social media?
I think the people really underestimate the power of LinkedIn. So, for example, before I joined this company, I did a self-training, you can call it. Like, for example, I trained myself. Before getting any job, I was just good at LinkedIn. I approached a lot of people on LinkedIn, and people are very friendly these days. You can write to a random person on LinkedIn and ask them for job advice, and you will be surprised that you get a phone call or a coffee with such people. So that's what I kept doing, networking a lot with people. And I developed a lot of skills how to do marketing on LinkedIn. And also, if you know how to do marketing on Google or Instagram or Facebook or Twitter, even some basic marketing skills, if you have, it's like really a plus point that you can use such skills for marketing on these platforms. As I mentioned, LinkedIn, Twitter, Instagram, Facebook, for your company you join for such a job. So it's very useful. Thanks a lot for pointing out, Felix. Such an important question.
Okay, Karim, I think we have answered everything. It was very informative. Thank you very much. We move now on to Dora, and she's a principal scientist at AHEAD Bioscience. And yeah, we are also very happy to have you. And please state your questions in the chat. You can also write it directly to me, which was happening now. And please be aware that we have a feedback questionnaire, and please fill it out. We want to make our events even better. Dora, thank you very much. The stage is yours.
So hello, everyone. Thank you, Felix, for the nice introduction. So I will start my presentation. I also would like to confirm with you if you can see the slides.
Perfect.
Okay, perfect. So yeah, let's jump directly in. So first I prepared a quick agenda of my talk. So I will walk you through the different stages of my professional career and also talk a bit about the motivation. And I will try to explain in each phase the learnings and expertise and also some of the learnings and tips at the end. But just to summarize, basically from the beginning of my studies, my main interest was to help improve patients' lives. So throughout my career, using different model systems and using different experiences, I tried to help improve different neurological disorders. So now to start with the path. So when I started preparing this talk, I decided, okay, let's lay out this career path. It looks very linear, very conventional. But then I was thinking, was this actually the reality? Was it this linear and this straightforward? So then I thought, okay, let's ditch this approach and actually focus on actually how it was, which was more like of a winding road. And I would like to give you some insights, not only on the professional aspects, but also on some hesitations, different questions regarding career path switch that came along the way. So for starters, I got my master's and bachelor in pharmacy in 2012. And after that, I spent 1 year working as a pharmacist in a community pharmacy. However, during this time, I already thought that this path was somehow too static for me, and I needed something more dynamic, as I was a very curious person. So I started looking around for opportunities, and this was when I called it here the conventional or comfort zone disrupting agent started, lurking around, and I started looking for research opportunities. And this is how I ended up then in Vienna in 2013 to start my PhD studies. My PhD lasted for almost 5 years. It was not always easy, but I learned a lot of things. And in the end, in 2018, I graduated. And again, after this PhD phase, I felt like, okay, my batteries were low. And I was thinking whether I should directly go into the next job or go directly for a postdoc. And this was again when this conventional zone disrupting agent started bugging me. And I thought, okay, let's just do what I feel like doing now. And I went for a 6-month break to recharge. And after this break, I started applying for jobs. And that's how I started then my postdoc in 2019 in Vienna at the Children's Cancer Research Institute. And shortly after being in the postdoc, I started wondering about my future and about my career, whether I really want to stay in academia or I want to do something more applied. And that was then the moment when I started looking into the industry and ended up at the biotech startup where I am right now. So this is the brief overview. I will go into detail of each phase. So as I mentioned, I studied pharmacy in Cluj-Napoca, so I'm originally from Romania. And in the next few slides, I would like to highlight the education and expertise I got out of this degree. So since it was pharmacy study, there was a high focus on chemistry and pharmacology expertise. Also, this was the first time when I got in contact with a lot of lab work and also with experiment-based approaches, which then, of course, later on will help me out a lot in my future endeavors. Also, since I was already curious about different research topics and projects, I already applied back then for a summer internship. And that's how I went then in my first abroad experience in France to do a internship in the medicinal chemistry department. In addition, since I was always interested in giving back to society and helping patients, I was also involved in a lot of public health campaigns, which also helped me interact with people and develop communication skills. And after the graduation, as I already alluded to, I started working in a community pharmacy, but I also wanted to further specialize in clinical pharmacy. However, due to some unlucky circumstances, I missed the deadline for confirming my spot for this clinical pharmacy residency. And given the fact that I was feeling not challenged enough in the pharmacy, this was the moment when this wind of change was coming around, and I thought, okay, how can I do to have a bigger impact on patients' health? Because in the pharmacy, I can only impact a limited number of people. And that was the moment when I decided to look for research opportunities. And with that, I relocated to Vienna to start my PhD in 2013 at IST Austria in neuroscience. And here on the right side, it's just depicted the cover of my PhD thesis, where I focused on autism and autism mouse models. So just again, a brief overview of the learnings from this phase of my profession. Regarding the environment at IST, there were three main pillars that were very important and helped a lot with my future development. First of all, IST Austria environment is very interdisciplinary. So this meant that in my first year, I had to take courses that were outside of my area of expertise and automatically outside of my comfort zone. So I learned, for instance, physics and linear algebra, which was a good training to just learn and prove yourself that you can do things outside of the comfort zone. Then the environment was very international, which again was very helpful to understand different perspectives and to see different ways of tackling problems. And as I mentioned, we did lab rotations, and later on, I also got the chance to be a teaching assistant for some courses. So it was a very a large variety of skills that were developed. And in terms of the specific expertise, so since my PhD was in neuroscience, of course, I got a lot of experience in neurobiology and stem cell biology, but also with working with different animal systems. I spent a significant amount of time with extensive experimental-based research, which helped me a lot then in developing a lot of transferable skills, also to do problem solving, because experiments don't always work out as planned, and also further enhance my analytical thinking. As I mentioned briefly, I also got the chance to teach students, but also supervise students in my last years of PhD, which then also opened up an avenue for different skill sets and teaching and communication. Also participated in collaborative PhD programs, such as the MOTAC program, which was actually very enriching, because you got to see different projects and different points of approach to tackle different problems. And of course, I also participated in different conferences where I had poster presentations, which again helped me with my communication skills and also a lot of networking. And also, again, coming to the part of giving back, I got involved in organizing a scientific outreach event, once for a young scientist audience, and also for the Science Industry Days. So as you can see, I highlighted all these activities just to enumerate some of the skills and expertise that I got out from such endeavors. And just briefly coming to the PhD project, so you understand basically my background better. So I work with a mouse model of autism spectrum disorders. And in this mouse model, the gene SRC7A5, which is a large neutral amino acid transporter at the blood-brain barrier, was basically knocked out. And this gene has been found mutating in patients with autism. So my goal was to try to understand basically the mechanism of how these mice might have a specific autistic phenotype. So I worked a lot with the molecular biology approaches. And here you can see with this mouse model, I looked at protein translation, which was decreased in these animals. I also did a lot of behavioral studies, and that's how I found that the social interaction was decreased in these animals compared to the controls, and also their motor activity was lower. And with the help of a postdoc in the lab, we also investigated neuronal network function, and this is how we found that inhibitory activity was decreased in the brain of these animals. And in order to attempt a rescue of this partial phenotype, of this motor phenotype, I delivered branched-chain amino acids directly into the brain of these mice by implanting mini osmotic pumps, and then basically doing what the transporter would do if it would have been present. And with this delivery, basically a partial rescue of the phenotype was achieved, paving thus the road of using amino acids as a potential clinical approach. And as I mentioned, these mutations were found also in patients, and basically there was a nice overlap between the phenotype with which the patients presented and also the phenotype that I observed in the mice, such as the autism, the motor deficits, and then the microcephaly, which was then later on confirmed with another model. Okay, and after I graduated from my PhD, there came the so-called transition phase. This was the moment when, as I mentioned at the beginning, my batteries were low and I was thinking whether I should continue directly into another job or just better take a break. So that's why I decided to take these 6 months off to travel, recharge, and enjoy life. And here's just a picture of me from those times when I went on traveling to Bali. After returning from my travels, I continued staying active in getting involved in different career development events. And this was also the first times when I actually came in contact with the ILSA at GmbH. And I also learned about these PhD awards that they were giving out for PhD thesis. And then I applied for this award, and then I was awarded the prize in 2018, which was also a nice token of appreciation for my work. And then I continued applying for jobs, and this process took longer than expected. So sometimes, of course, I felt bitter, and I started wondering whether taking this break was actually a good idea, although I had already maybe some offers to continue as a postdoc. However, I went through with this plan, and I think it was important because it helped me become accountable for my own decisions and also to learn from discomfort and trust the process in the end. So in the end, the 6 months kind of turned into 9 months. But in the end, I decided to continue with research because this was still one of my main drivers. And I wanted to expand my knowledge and slightly change research field. So that's how then I ended up with the postdoc at the Children's Cancer Research Institute here in Vienna. And basically during this time, I still focused on the nervous system, however, from a cancer perspective. So I worked on neuroblastoma, and this helped me also expand my molecular biology expertise in terms of working with CRISPR libraries. I've done a lot of single cell sequencing, and also I worked directly with patient samples. Again, this time was very fruitful because it enabled me to establish different collaborations with many research groups and also focus more on project management and having a more independent work style. However, thinking of the bigger scheme of things and wondering whether staying on the academic track was really something that I wanted to do, then I started wondering what to do next, or if I should continue on this path, especially because I always had this drive to not only answer my research curiosities, but also do something applied, something that can be used for patients or directly have an application in industry. So then through a serendipity, I came upon an announcement about a newly founded startup here at the Vienna Biocenter that was focusing on brain organoids. So I thought, given my expertise with neuroscience and stem cell biology, I thought, okay, let's just send an open application and see where this will take me. And this was then how I ended up at Ahead Bio as a senior scientist in 2020. So just a brief overview, Ahead Bio is a spin-off company from IMBA here in Vienna. And the aim is to develop new therapeutics for the treatment of brain disorders, making use of the brain organoid technology. So just to give you an overview of what we are doing and what our mission is. So we try to capture diseases from patients in human organoids. So what this means is that you can take blood samples or skin samples from patients, and then these samples can be reprogrammed into stem cells, so-called induced pluripotent stem cells. And then from these stem cells, basically one can derive any type of tissue, but in our case, we derive brain tissue in the so-called organoids, as you can see here in this image. And then we try to mimic then the biology of the respective patient with these brain tissues. And in order to inspect the relevance of the model and see how this translates in terms of functionality, we use network function, network activity, in order to evaluate the network in this organoid. And how we do that is that when we have the induced pluripotent stem cells, we genetically encode a optical sensor, and then this optical sensor is getting integrated into digital DNA. And once we generate organoids from these cells, when they reach a certain level of maturity, you can see in the video here on the right-hand side, all these flashing cells are basically neurons that are active and they are expressing this sensor. And this allows us then to evaluate the activity of the brain tissue that we developed. And coming to my responsibilities and tasks, since this is then one of the main focus points of the talk, I would like to give you an overview of what I do on a daily basis, keeping in mind that since it's a biotech startup, the environment is very dynamic, and you get to do all sorts of activities and tasks, and the responsibilities, let's say, are not fixed. So sometimes if there is a problem, you have to chime in and fix it. So definitely one important aspect of my daily work is let's say scientific reporting. So this can be done either internally when we have progress reports, or at a certain amount of time when we have to write reports to the funding agencies on our progress. Then I spend a fair amount of time with project writing and project management, making sure that we adhere to timelines, allocating resources, and distributing tasks. Since I'm still doing also experimental work, I also spend time doing literature review, designing experiments, trying out different protocols and optimizing protocols, and eventually also analyzing data that comes out of the experiments I have set up. Then I also participate at different scientific discussions, either internally or at seminars, or when we have different external collaborations, because this is then very important to come up with new ideas. And I also got the chance to supervise a great team of scientists, and we work together on advancing our common goal as a team, and then we can give feedback. I give feedback on ongoing projects, and we have, let's say, an open communication in terms of feedback. Then I also establish collaborations with research partners, depending on the types of approaches that we have going on at the moment. And lastly, but maybe more importantly, it's a troubleshooting, because as I mentioned, it's a startup, it's a very dynamic environment. So it can be that something doesn't work out. So you have to get hands-on and try to fix it. Okay, and in terms of skill set, I try to address some of the hard and soft skills that I consider are needed for this job. Of course, none of them, let's say, is a finite skill. It's always can be done better, and it's always a work in progress. So in terms of hard skills, definitely having my background in neuroscience and also experience with stem cell biology, 3D biology, molecular biology have helped me a lot to get this job and also to be able to do the work I'm doing. Then also a series of microscopy techniques that I have learned during the PhD and then also during the postdoc were very helpful. Project management skills, which I have developed mainly during the postdoc times, were further improved during my job here. And then also biostatistics knowledge for doing data analysis and also presentation skills. I think those were some very important hard skills. And then to balance the hard skills, of course, there's the set of the soft skills, which again, as I mentioned, they are always work in progress. Some can always be improved or done in a different way. So in terms of soft skills, definitely proactiveness for a startup environment is crucial because sometimes there are problems and you just have to go for it and try to fix it and be proactive on finding answers and solutions. Teamwork is another very important aspect because we work collaboratively. So we have individual projects, however, the projects get intermingled. So it's very important to have trust in each other and also to contribute. Because if one of us, let's say, cannot contribute or does not contribute, then the other ones have to back that up. So we really have to have this fluid teamwork. Then adaptability, of course, because of the changing nature of the environment is very important, which comes also with accountability. As I said, if you go out and try to fix something or have a solution, then you also have to be accountable for the outcome. And then one that I really is a fine balance, let's say, between hands-on and hands-off approach, especially when it comes to supervising a team. I think this is always important to know when to give people more freedom and to allow them to experiment on their own and find solutions. But then there's also times when you have to maybe get more hands-on and take responsibility or have to make some decisions. Critical thinking, which having done a PhD and a postdoc, of course, they were there, but it can always get expanded. Don't take things for granted. Ask your own questions. Go out and experiment. I think this is very important. And of course, communication, which goes hand in hand with teamwork, maybe, but also communication with research partners, I think is very important. And in addition, I also added a few other skills that, at least for me, I think were very important also to improve in my job, but also to get to know other people. So, for instance, taking a leadership course, especially in the light of having to supervise people, this was very useful for me. And also networking with other scientists and other associations. I think we have quite some opportunities here in Vienna. So I really encourage you to try to network. There are regular events from GmbH, from Austrian startups, from the Health Hub Vienna, and from LISA Vienna. So I think these are all very helpful, and you can only learn things. Okay, and for the last few minutes, a few learnings and tips that applied to me, and I would like to share with you. You can see if it fits or not. So know yourself and know your values. So for me, this is a very important aspect because as I have gone through my professional life so far and different stages of life and career, sometimes needs or desires change. And it can even happen that maybe when I set out on this path to study something, it was not even me, but maybe it was sometimes teachers or parents or society who initially set you up on a path. But it doesn't necessarily mean that you cannot switch directions or do it in your own way. So you don't have to follow, let's say, the conventional path. And that's why I think for me it was important to have a set of values, to know when to make compromises, And here I put like a funny graph about this yearning octopus, which I think we all have kind of inside of us, right? Because there's always different needs that we have, be it personal, social, moral, practical, and in different stages of our lives, we might prioritize some over the other. So I think it's good to look at this and maybe even use this tool that I offer you here to try to find out your values and maybe come back and do it again in 1 or 2 years and see what changed and what stayed the same. And then this can help you also in making your decisions in terms of career path. And the second message would be to build your path as you go. So for me, this was actually very important and liberating to know that you don't have to follow something that was laid out, let's say. And using this kind of growth hack approach, I think it's very important to experiment and sometimes take a leap and try out things, even though it might be uncomfortable sometimes or scary. But if you're a scientist, you're anyway used to doing experiments. So I think you can apply that also to your daily life. And then you can measure the outcome of these experiments and you can learn from your mistakes. And also try to focus on your own story, maybe not compare to other people, because this is also often, let's say, a source of bitterness. And then once you learn from this and accumulate these insights, then you can optimize and use second order thinking and wonder, okay, so if I do this, then what, and so on. And then you go out and you experiment again. And yes, it can feel like jumping out of the plane, which for me was the case. However, you can do any kind of other experiment, whatever floats your boat. But I think the important part is to know that you can make your way as you go. And for the take-home messages, so yes, hard skills and professional degrees are important, but I encourage you to dare giving them a personal or unconventional flair. Stay curious, proactive, and try to get comfortable with your peculiarities. So maybe if you have some weirdness or something that you think is a weakness, actually you might use that as a unique selling point. And lastly, experiment with your limits, with your beliefs. Try to welcome your own type of conventional zone disrupting agent. And also keep in mind that you have to go out there and get what you deserve. Sometimes things will not come to you just because you deserve it. So sometimes you have to go out and get it. So with that, thank you for your attention. And yes, I'm happy to take any questions.
Dora, thank you very much. This was very, very nice. I'm intrigued. Also, the personal insights was very great, very great. I got the question, what is the biggest difference between academia and now the startup in terms of doing science?
Okay. Yeah, so definitely there's still a lot of research. So let's say in terms of daily work with doing experiments, this is still the same. However, there is, let's say, higher emphasis on reproducibility and robustness of processes, right? Because with academia, of course, you might repeat an experiment 3, 4, 5 times, and then you have the paper, and then that's that. But here, you really have to have a robust, reproducible process in order to go out and claim a certain, let's say, a phenotype or something. So I would say this is one of the big differences.
Mm-hmm. What's in terms of over hours? Is it like more strict in your field and industry, or would you say it's also necessary and you do it a lot?
Yes. So in general, with startups, there are these all-in contracts. So you have a certain number of over hours that you do, and you're kind of covered in your contract. But let's say it's like 4 or 5 hours a week. So it's not maybe 60 hours or 50 or something that sometimes happen during the PhD.
Yes, true. You said you're still in the lab doing microscopy and so. What's the ratio lab work and being in the office approximately?
So I would say it changed at the beginning, since I'm here for 3 years. Since at the beginning we were fewer people, then I would say it was maybe 60% in the lab and 40% office. Maybe now it's the opposite, because now there are more people, and then I spend maybe 60% on researching, writing reports, project management, but still I keep 40% for experimenting and trying out different protocols.
How big is your company?
So we are currently 11 or 12 people. We have some people that are on maternity leave, but yeah, in total we are 12.
12 people. There is a question from Veronica. She would like to ask about the financial aspect while doing a PhD. Is it demanding? I think she's referring now to the 60 hours and the ratio what you get. Maybe Veronica, you can tell us if this is true. Yeah, is this the question? You can also speak up. I think this is also possible.
Yes, I wanted to know how it basically works, because it's pretty time-consuming PhD, I'm sure, and maybe some advice for the future, or how it was.
So in terms of financial aspects, you mean now, or?
Yes. That was the question.
Okay. So basically, yeah. So after the PhD, I had the postdoc. So then you have this automatic increase from the FWF. I think they have these rankings for the different salaries. And then coming with this postdoc experience, then I had the same, basically at the startup, it was like continuation of the postdoc. So I stay, let's say the ranges are, even startups use those FWF ranges for the salaries based on the experience. Yeah. So I came with the postdoc experience.
You said something about you have to balance hands off, hands on. Can you emphasize this a little bit more? I'm interested. You should not get stuck by a problem, or what do you mean by that?
No, so this I meant more in terms of team supervision, right? Because at the beginning, when you have maybe new people to supervise, it's okay to be maybe more hands-on. People need more guidance, need more feedback, more input. But then, of course, yes. And as time goes by, then people, of course, because we're all scientists and are curious and have our own hypothesis, then people need more freedom to test out their own hypothesis. So then maybe it's more hands-off and only get feedback or discuss at a certain time, but not so often.
I see. Okay. Great. Thank you very much. There are no other questions.
I think there was something from Amelie. Sorry, I just see now in the chat because I opened.
You got it directly.
I don't know if this was answered.
No, but it's a personal question for you.
Okay. Sorry, I just saw it. Yes.
Okay. Yeah, great. I thank the speakers very much. It was very, very great to get these insights.